Very good evening, my dear friends. So today, our eminent resource person, Dr. A. Madhu Madam Garu, Assistant Professor, Department of Microbiology, Government Degree College for Women, Begumpet Autonomous, Hyderabad. Madam is going to explain today's one of the important topic in our CPGET Botany Plant Sciences, that is genetic code. So, Madam, please start the session.
Good evening, Amma. Today's topic is genetic code. So let us understand genetic code. What is genetic code? Okay. So it is a dictionary that corresponds with the nucleotides and sequence of amino acids. Like if we want to write something, if we want to talk something, we have a language. Okay. So genes are also having some language to express their information, right? So genetic code is a set of rules by which information encoded in genetic material is translated into proteins. So where this genetic code is helping to translate or to convert the language of nucleotide sequence into sequence of amino acids. And the term genetic code is given by the scientist George Gamow. These are the people who have worked on this concept to understand how proteins are encoded began after the structure of DNA was discovered. Watson and Crick has given DNA structure. From that, we can understand how these genes are getting translated. George Gamow postulated that a 3-letter code must be employed to encode the 20 standard amino acids. So we are having how many amino acids in nature? 20 amino acids. So to code or to produce that amino acids, we need some code. Okay, that is 3-letter code is required to make these amino acids. Next is introduction to genetic code. So like we are talking, we have many words in any language, right? This genetic code or the language of genes is having letters. We are having ABCD in our language, English language. Similarly, this genetic language is having 4 letters. What are that letters? Adenine, guanine, thymine, and cytosine. And the collection of codons is called as genetic code. So these codons are made of which letters? A, T, G, C letters, right? And how many amino acids are there? 20 amino acids. So for 20 amino acids, 20 codons are sufficient, right? But is this the picture in nature? No. Each codon, there are more than 20 codons. Let us see how. Each codon should have 3 nucleotides to impart specificity to each of the amino acid for a specific codon. Now, genetic code is a triplet code. We call it as a triplet code. Why we are calling it as a triplet code? Because 3 bases are there. 3 bases make 1 codon. Okay. How it is proved is, if we take 1 nucleotide, how many bases, how many nucleotides are there? 4. So, if we take 1 nucleotide to code 1 amino acid, how many amino acids can be coded? Only 4 types, 4 combinations. If we take 2 nucleotide combinations, okay, like AG1 combination, AT1 combination, AC1 combination, like that also if we take, we get only 16 combinations. But how many amino acids are there? 20 amino acids. 20 amino acids have to be coded by this code, these letters. Okay. So next option is 3 nucleotide language. Okay. If you take a set of 3 bases. Okay. So how many combinations are possible? 64 combinations. Here you can take it as 2 to the power of, we are having 4 bases. So 4 to the power of 1 is 4. 4 to the power of 2 is 16. 4 to the power of 3 is 64 combination. So this number 64 is definitely higher than 20, which is sufficient to code for all the amino acids. But again, the question arises, only 20 amino acids are there. So why 64 combinations? We have understood if we have less combination, like if we take the combination of 2 nucleotides, it is not sufficient. If we take the only 1 base as a combination, it is not sufficient. So definitely it should be a triplet codon. Okay, so in this slide, what we are understanding, genetic code is a triplet code. See, this is 3 bases, 1, 2, 3 bases. This makes one codon. It will code for one amino acid. This is most important concept. Okay, one codon. One codon is made of 3 bases. It is coding for one amino acid. Next, we are having this table UCAG. What are these? These are bases. UCAG. These are bases. Okay. Here, if you just pay attention, we have written uracil. We have written uracil. Uracil is present in DNA or RNA. It is present in, yes, participants. It is present in RNA. Uracil is present in RNA, not DNA, right? So we are taking combination of bases present in RNA and we are checking the combination. Okay. Here, how you have to write, this is the first position. This is the second position. Like in the last class, we have written the Punnett square. Similarly, here you have to write this combination, right? So U. First position, U. Second position, U. Leave the third position. So all the 4 bases, first and second position is UU. And what about the third base? You write here in the sequence, UCAG. Similarly, second box, first position C, second position U. CU, 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 CU. Then UCAG. So likewise, you will get all the 64 combinations of codons, which we can get by using these 4 bases, that is U, C, A, and G. Okay, so like this, you have to write all the 64 bases. And then what we have to do, if we have to remember this, which codon is coding for which amino acid. This is coding for phenylalanine. These 2 are coding for leucine, and these 4 are coding for, these 2 and these 4, total 6 codons for leucine. These 3 are for isoleucine. AUG, this is a special codon. That is why it is shown in color. Okay, this is a initiation codon. Okay, that is coding for methionine. AUG is always an initiating codon, right? First codon will be AUG. Then these 4 are coding for valine, serine, proline, threonine, alanine, tyrosine, and these 2 codons are stop codon. This is also stop codon. You have to remember the sequence. AUG is for methionine. It is an initiation codon. These 3 are stop codons: UAA, UAG, and UGA. These 3 are stop codons. You have to remember these sequences. It can be asked in the exam. Next, histidine, asparagine, lysine, aspartic acid, glutamic acid. Likewise, all the 20 amino acids. If you count the number of amino acids, you will get 20 amino acids in this table. Okay, so this is first position, this is second position, and this is third position, right? Likewise, you have to make this genetic code table. And as we have discussed, it is a dictionary. It is having all the codons. Each codon is a triplet code. Total, there are 64 codons. Out of the 64, 61 are coding for amino acid. Just check. These 3 are not coding for any amino acid. Means if in 64, if you remove 3, then it will be 61. So 61 codons are coding for amino acids. And 3 codons do not code for any amino acids. They are called as termination codons. Now, whatever codons we have seen, there are 2 types: sense codons and signal codons. Signal codons, here you have to understand what are signal codons: start codon and stop codon. Why we are calling them as signal? Because they are giving some signal to the process. Whenever we are finding AUG, that is a start codon. Okay, and whenever we find stop codons or termination codons, that is a signal to stop the process of translation, right? So that is why they are called as signal codons. And apart from that, all are whatever codons are there, 61 codons, they are called as sense codons, right? Means they are having some sense. And these are the termination codons: UA, UAG, and UGA, and AUG is a start codon. Next. Just now we have seen this. They have given some names to this. You have to remember. UAA is amber. UAG is ochre. UGA opal. It can be asked. Okay. These three are termination codons. The names given to these codons are amber, ochre, opal. Right? Initiation codon, we know AUG. It always codes for methionine. In prokaryotes, it is coding for formal methionine, and in eukaryotes, it is coding for only methionine. Important difference. Okay, important difference in translation is in prokaryotes, AUG is coding for formal methionine. In eukaryotes, it is coding for only methionine. Next, what is anticodon? What is anticodon? Till now, we are talking about codon, codon, codon. Where is this codon present? Yes, it is present on mRNA. Codon is present on mRNA. Then what is anticodon? The opposite sequence, which is present on tRNA, is called as anticodon. Here, it is very important to understand what is codon and what is anticodon. When you are talking about codon, we are talking about the sequence present on mRNA. That is why we have written uracil. We have not written thymine. If it was a DNA sequence, we would have written thymine, right? But we are not writing thymine. We are writing only uracil. So it is an RNA sequence. RNA sequence means particularly it is mRNA sequence because if you go back to basics, right? How the information present on DNA is converted into proteins through mRNA. Okay, the information present on DNA is copied onto mRNA, and from mRNA we will be getting proteins or amino acids, right? So the codon is present. It is a sequence present on mRNA, and anticodon is a sequence present on tRNA. This is an important concept you have to remember, right? This is the anticodon. This is the codon, and this is a wobble position. Wobble position means it can change. It can change. Okay. So that third position is wobble position. And difference between codon and anticodon. Just now we have seen codon is present in both DNA and RNA, but anticodon is present in RNA and never in DNA. But now whenever we are talking about translation, codon is always present on mRNA. Okay. Now, codons are written in 5' to 3' direction, whereas anticodons are usually written in 3' to 5' direction. Okay, if you have studied replication, right? What is the direction of replication? Can anybody answer? What is the direction of replication? It is always 5' to 3'. Okay, important bit. Direction of replication is 5' to 3'. Transcription is 5' to 3'. Direction of translation is 5' to 3'. This is important. Okay, all the processes are in which direction? 5' to 3'. Why we are talking about this? Okay, codons are always written in 5' to 3', whereas on anticodon direction is 3' to 5'. Now, anticodons of some tRNA molecules have pair with more than one codon. Anticodons are present on tRNAs. tRNAs are carrying amino acids. Okay. Now, how many amino acids are there? How many amino acids are there, ma? Number of amino acids? Please, you have to interact. If you interact, I can understand you are following me. Yes, ma? Anybody? What are the number of amino acids present?
20, ma.
20. Very nice. Okay. So if there are 20 amino acids, we have seen the justification, like why there are 64 codons or 61 codons. We have understood it. More number of codons are there because combination with two bases is not sufficient, right? But 20 types of amino acids are there. So 20 types of tRNAs are there. Here one more concept is there. tRNA's number is equal to the number of amino acids. Am I clear? tRNA's number, types of tRNA number is equal to the number of amino acids. Means if methionine is there, one tRNA is there, right? If alanine is there, that is also having one tRNA. Means what these tRNAs are doing? What is the function of transfer RNA? tRNA means transfer RNA. What it is transferring, where it is transferring. Anybody? Kavya, Swapna? What is the function of tRNA? Yotsna? It is transferring. T stands for transfer, right? It is transferring amino acids. From where? From the cytoplasm to ribosome. From cytoplasm to ribosome. Okay, if we are constructing a wall, right? We need bricks. We get from somewhere to the construction site. That bricks, right? Similarly, here tRNA is transferring amino acids from cytoplasm to ribosome where the protein synthesis is occurring. Okay, so the number of tRNAs is always equal to the number of amino acids. So number of tRNAs here is 20. Number of amino acids is 20. Number of codons is, what are the number of codons? 60. Definitely it is more. So here you have to see anticodon of some tRNA molecules have to pair with more than one codon. Is this statement justified? As the number of codons are more and tRNA molecules are less, they have to pair up with more number of codons. That is the concept here with the wobble base. Wobble base means this base can code with a different combination. Okay, these two bases are important for pairing. This base is having some flexibility. Am I clear here? Next, codons are arranged in nucleic acid strands with anticodons are discretely present in cells, amino acids attached or not. So what is the role of codons or sequence of DNA? Okay, it shows the sequence of amino acids. Okay, let us suppose there is a sequence on DNA which should code for methionine, alanine, phenylalanine. So that language is translated into the similar sequence. When it goes for translation, definitely first comes methionine, then alanine, then phenylalanine. So the codon or the language present on DNA is it is dictating the direction of amino acids to be present in a given protein. Anticodon helps in bringing a particular amino acid as its proper position during translation. So anticodon is present on tRNA and tRNA is carrying amino acid. These are the characters of genetic code. Am I audible? Okay. So what are the characters? It is a triplet code. Triplet code means 3 bases. It is commaless, means after 3 bases there is no comma, or after 4 bases there is no comma. There is no stoppage. Okay, it is continuous. And it is non-overlapping. Non-overlapping means in the coming slides we may get it. Code is not overlapping. First 3 bases it will take, then next 3 bases, then next 3 bases. Code is never overlapping with each other. Coding dictionary, we have seen that 64 codon, and it is a
Madam, your voice is very low, madam. Excuse me, madam. Excuse me, madam. Madam.
Am I audible?
Yes, madam. Now it's audible, madam. Please proceed.
ఎక్కడ నుండి వినపడలేదు?
ఒక 1 minute నుంచి వినపడలేదు.
ఓకే ఓకే. So code degeneracy means one amino acid is coded by different codons, many codons, more than one. That is code degeneracy. And universality of code. Genetic code is almost all same in all the organisms. Some exceptions are there. And it is a non-ambiguous, no difference. And chain initiation code, chain termination codons. Codons, we have seen initiation codons and termination codons are also there. Okay. And this we have seen before also. This is code degeneracy. Like this amino acid leucine is coded by how many codons, ma? Here 4 codons are there and here 2 codons. Okay. So overall how many codons? 6 codons are coding for leucine. That is single amino acid. This is called as code degeneracy. Am I clear? Whenever the voice is not clear, just stop me and you can ask. Right. Universality. When we are saying it is universal, it is present in all the organisms. What are some exceptions here? AUG is the codon for methionine in mitochondria. The same codon codes for isoleucine in cytoplasm. This is the exception. Okay. When we are saying AUG is always a initiation codon and it is always coding for methionine. But where is the exception? In cytoplasm, it is coding for isoleucine. Non-ambiguous. Non-ambiguous means the one codon cannot specify more than one amino acid. Okay, we are talking about two concepts here. One is code degeneracy and the other one is non-ambiguous nature. Code degeneracy means single amino acid coded by many codons. Single amino acid coded by many codons. Non-ambiguous means one codon cannot code for more than one amino acid. These are two opposite concepts. Okay. Next, non-overlapping. Non-overlapping means if you write some words or letters, okay, A, G, C, T, G, C, okay, A, G, C is one codon, T, G, C is one codon, okay, it can never be after A, G, C, the second base cannot form next to codon, okay, after 3 bases, 4th base can only start the next codon, not second or third base. Understood? It is not overlap. Next, continuous translation. Gene is transcribed and translated continuously from a fixed point, a fixed starting point to stop point. Punctuations are not present between the codons. If we are writing a sentence, if we are writing a paragraph, right? We put commas, right? We put commas. Or punctuations will be there. But in genetic language, there are no commas. There is only a full stop. Okay? There is a start point, there is a stop point. There is no intermediate stopping between these 2 points. Now, code has polarity. Code has polarity. It has definite direction for reading a message, which is referred to as polarity. When we are saying replication is in direction of 5' to 3', there is a direction. There is a polarity. So definitely this polarity will be there in every stage of this processes. Reading of message from left to right or right to left will specify for different amino acids. For example, UUG stands for leucine. From right to left it is GUU, which stands for valine. When you are saying it has a polarity, it means you are saying that there is a particular direction. Why there should be a particular direction means the same message, if you are reading from left to right, it is giving one amino acid. Same message, if you are reading from right to left, it will give another amino acid. So this is the reason why there should be a particular direction to the genetic code. Next, degeneracy, we have already discussed. Single amino acid can be coded by more than one codon. That is code degeneracy. And wobble hypothesis, we have discussed little bit in the just before. So what is this wobble hypothesis? It accounts for degeneracy of genetic code. What is degeneracy? Degeneracy means one amino acid is coded by more than one codon. Okay, so how it is possible? How it is, how the single code or many codons are coding for the same amino acid. See here, leucine. This tRNA is carrying which amino acid, ma? Leucine. This is also carrying leucine. Okay. So what is the codon here? CUC. What is the anticodon here? GAG. This is the normal pairing. This is the normal pairing because see, the hydrogen pairing, that pairing is between which bases? AT and GC, right? So if there is cytosine here, definitely there should be a guanine. Here, thymine, in place of thymine, we are having uracil in RNA. So it has to pair with adenine. And here we are having cytosine, so cytosine, it is pairing with a guanine. Okay, so this is normal pairing. Now, let us see here, same codons for two positions, CU, and the third position is having uracil. Third position is having uracil. But the tRNA is same. The tRNA is same. The anticodon is same. But here codon is different. But CUU, what it should code? CUU, just see. Where is this? CUU, ma? Cytosine. This one. CUU. This is also should code for leucine. So what is happening here? tRNA is same, codon is little changed. So the third base, it is showing flexibility in pairing. That is wobble pairing. Actually, guanine should pair with cytosine, but even if there is uracil, it is pairing with this base. So this, the flexibility at the third position of codon anticodon pairing is called as wobble pairing. This is responsible for degeneracy of code. Hope I am clear. Okay, so that is why this degeneracy is there. And the biological significance of degeneracy. Biological significance means, here you can explain like if there is a mutation in a cell. Okay, by mutation, some base sequence or some at single point one base is changed. Suppose this same example we take, okay, there was a mutation in this sequence and this cytosine has changed to uracil. Now what happens? What happens if it normally, if there is no codon degeneracy, if there is no wobble pairing, definitely this codon will pair for different amino acid. Yes or no? If it is coding for different amino acid, protein is different. If the protein is different, functioning is different of that protein. So ultimately leading to some biological problem in that person or in that individual, right? But here, same. When this degeneracy is allowed here, it is coding for the same leucine. Okay, due to this wobble pairing, same leucine is being coded. So there is no change in protein and no change in the final function of the protein. That is the biological significance of degeneracy of the genetic code. If there is a mutation by mistake, okay, and due to that mutation, if it is coding for the same amino acid, which is allowed by this code degeneracy, that is protecting the individual from deleterious effects of this mutation. That is the significance, right? Flow of genetic information. This is the basics, okay? From DNA, you have to get RNA. From RNA, you have to get protein, right? This is DNA sequence. From here, you are getting RNA. From RNA, you are getting peptide or protein. First, you will get peptide, then it will be converted into protein, okay? Next. How much time is there?
Madam.
Okay.
Madam, 8 minutes. Madam.
Okay.
8 minutes left, madam.
Okay, okay, sir. So mutation when explained using genetic code. Silent mutation. Silent mutation means just now we have seen if the base is changed here, the base is changed. There is a mutation, there is a change in base, but this A is changed to guanine. Adenine is changed to guanine, but both the codons are coding for the same amino acid. So there is no effect in the protein. That is called as silent mutation. Hope I am clear here. Missense mutation. Missense. Sense is changed. Sense is changed. Let us see here adenine. Adenine is, adenine is changed to cytosine. If you compare the sequence, adenine is changed to cytosine. So CAT is one codon. Here CCT codon. CAT was coding for histidine and CCT is now it is coding for proline. So amino acid is changed. So the protein will be changed. That is there. It is called as sense. Means the sense is changed. The protein is changed. Nonsense mutation. Nonsense means termination codons or stop codons are also called as nonsense codons. Okay, they are also called as nonsense codons. So just to see here, cytosine is changed to thymine. Cytosine is changed to thymine. Here, this codon is coding for glutamine. Here, it is coding for stop codon. That is it is coding for like it is not coding for any amino acid. That is the stoppage of this peptide synthesis. Okay, it is not coding for, like when you are talking about stop codon, you have to remember stop codons will not have any amino acids. Okay, they won't code for any amino acid. That is why they are called as stop codon. And this TAG sequence, it is represented as UAG in RNA, right? Frame shift mutation. If this is deleted, okay, if this is deleted, the system of replication or transcription or translation, they cannot understand one base is deleted here. It will consider the sequence as continuous. Okay, so the sequence will be changed to ATG as it is. Then GAA is changed to, as G is not here, AAG, then CAC, then GT. It will continue, right? Understood? This base is missing. So it is taking the sequence in a continuous fashion. ATG, AAG, CAC, GT, etc. So the sequence of amino acids should be methionine, glutamine, alanine, glycine. But now the sequence has changed to methionine, lysine, histidine, which is a different sequence. Here, this should be the sequence, but here this is the sequence which is changing the entire sequence of amino acids from this point where the mutation has occurred. This is called as frame shift mutation. Here you got some idea about mutations also. Hope this will help you in solving the problems related to genetic code and some mutation problems. Okay. Now, if you have any questions, I'd like to answer. Actually, I wanted to solve some bit, but it took more time. So next class, definitely, as holidays have extended for 3 more days, we can cover all the genetics and all bio bits. Also, we can try. Okay, sir. We'll wind up the class.
Okay, madam. Okay, madam. Thank you very much. So, my dear friends, today's excellent class we have listened, and definitely this is one of the most important topic regarding of our CP GET. So many questions posed from this topic. So, and also, my dear friends, this video is will upload in our YouTube channel also. So, my dear friends, tomorrow also we have sessions. Follow a WhatsApp group and join without fail. Thank you, madam. Thank you very much. So, I will going to wind up the session.
Thank you.